Determining if bacteria are motile can be helpful in identifying them. Flagella are the only organelle for motility in prokaryotic cells, but the flagellum is only a single strand of protein, much too small to be seen with a light microscope. However, if we can demonstrate the organism is motile, then we've demonstrated that it has flagella. There are three ways to demonstrate flagella. One is using a flagellar stain. This is helpful if you need to know the number and arrangement of flagella. Usually this is not that important. A flagellar stain is very demanding and takes a lot of time to do. A hanging drop is another microscopic procedure. To do a hanging drop, you put a drop of liquid culture on a cover slip and then suspend that over a well on a microscope slide. You look to see if the organism makes progressive movement. This can be a little challenging because bacteria are very small and they're unstained, so it's hard to see them sometimes. Probably the simplest way to determine bacterial motility is to use SemiSolid Auger. The SemiSolid Auger technique involves selecting colonies for testing using a sterile needle. You stab the needle halfway down into a motility media deep. So here you see just halfway down. Then the tricky part is coming straight back out, no lateral movement, no wiggling whatsoever. You then incubate this tube for 18 to 24 hours at 37 degrees centigrade. After incubation, you look at the tube. Motile organisms will be able to swim through the auger and leave a cloud of turbidity behind. Non-motile organisms will grow only in the stab line, leaving a nice sharp stab line. If you add a tetrazolium salt to the medium, it makes visualization easier. Tetraphenyl tetrazolium chloride, or TTC, is colorless and soluble in its oxidized form, but it turns red and insoluble when it's reduced. Flagellar movement and other metabolic activities require ATP. When ATP is broken down, electrons are released. TTC can pick up the electrons and become reduced TTC. That means that any area with metabolic activity will appear red. So here you see a positive motility, a nice cloud of growth, no clean stab line, and here you see a very clean stab line. This activity at the top is because there was moisture on the top of the auger. That allowed the organism to spread around on the top, and sometimes it can get down between the tube and the auger if there's moisture there. So you want to look here at the tip of the stab line. Nice and clean and sharp, no movement. Over here, very fuzzy.